Multicolor microscopy images contain chromatic shift in 3 dimensions that impair colocalization analysis of high-resolution images. It is generally difficult to remove chromatic shift originated from a biological sample itself. Our method can correct chromatic shift in the biological sample using separately acquired reference images. Demonstrating the procedure will be Takako Koujin, a technician from my group.
Begin by seating 2.5 times 10 to the 5th HeLa cells on a 35 mm glass bottom dish and growing them in a carbon dioxide incubator. After 24 hours of incubation, replace the medium with 2 milliliters of 3.7% formaldehyde in PBS and mix gently. Fix the cells for 15 minutes at room temperature on a rotating platform. Wash the cells 3 times with 2 milliliters of PBS for 5 to 10 minutes. Permeabilize them with 2 milliliters of 0.1% Triton X100 in PBS while rotating, and repeat the washes with PBS. Then stain the cells using a standard antibody staining protocol as described in the text manuscript. To obtain a reference image of crosstalk reference for wide-field microscopy, add 2 milliliters of 0.5 micrograms per milliliter DAPI to stain the DNA. Leave the cells on the rotating platform for 30 minutes. Then replace the solution with 100 microliters of mounting medium. To obtain a reference image of biological calibration reference for any